Alright, so this unit is on cell reproduction, and there's actually two specific things we'll be talking about throughout this unit. The first is asexual reproduction, or mitosis, by which cells make exact copies of themselves. And the other one is sexual reproduction, or meiosis, which is the process by which cells cut their chromosome number in half to make gametes, or sperm and egg cells. So, first, a little introduction. Why do cells reproduce? Well, there are three main reasons why cells need to reproduce. Number one, it allows an organism to grow. If you think about it, you started as a single cell, and now you're made of millions of cells. So obviously, in order for you to grow and develop and become a full human being, your cells had to divide. Secondly, to reproduce cells that become damaged or age and die. Some cells in your body live as long as you do, such as brain cells and muscle cells. Those cells really don't reproduce. They stay alive, they're born or form when you're born, and you have them your whole life. But other cells in your body have a very limited lifespan. For example, skin cells only live about 35 days. Red blood cells live about 120 days. So you have to replace those cells, and so cell reproduction would be the way that's done. And finally, to make more organisms. When we reproduce sexually, or even when single-celled organisms reproduce, just by making exact copies of themselves, the cells have to reproduce in order to make new organisms. So, when a cell does reproduce, what is actually passed on? And the answer is the DNA. The DNA is passed from the original cell to the new cells. Some cytoplasm is passed as well. And I may have mentioned this before, but in eukaryotic cells, at least in animals, in humans, it is only the mother that passes on mitochondria. So technically your mitochondrial DNA, if you remember that there's DNA in the mitochondria, they all came from your mom. Your mitochondrial DNA would be identical to your mother's, and identical to her mother's, and her mother's, and her mother's. You can literally trace it back many, many generations, and you'll actually have identical mitochondrial DNA to the maternal side of your bloodline. If you're a male, you will never pass your mitochondrial DNA on. You have mitochondrial DNA, but you won't pass it on to your offspring. All right, but for now, we're going to be talking about, just to clarify, we're going to be talking about nuclear DNA, the DNA in the nucleus. So the DNA that you have in your cell, in the nucleus of your cell, is called your genome. The smallest cell in your body with a full genome would be a somatic cell, or a body cell. A sperm or egg cell would not have your full genome because sperm and eggs only have half of your DNA. So the genome can be one single DNA molecule, such as in prokaryotes, they have literally one loop of DNA. Or the genome could be carried on multiple DNA molecules. Like us, we have 46 chromosomes. So our genome, the genome of a human, would consist of a set of 46 chromosomes, or 46 DNA molecules. And a chromosome is basically a package of DNA. Technically, it's not just DNA. It's in the nucleus, in a eukaryotic cell, and it is DNA along with protein. And it's in a mass, normally, when the cell is not dividing, called chromatin or chromatin, depending how you may hear it pronounced either way. And chromatin is 60% protein, 40% DNA. In fact, when they first started studying genetics and they started trying to figure out what was the important stuff that was getting passed from parent to offspring, they actually thought it was the protein that was important because it was 60% protein. But it turned out it was the DNA. Alright, so what is the job of the protein? Because it does not have genetic instructions. Well, DNA has a negative charge overall. It gives off a negative charge. The proteins that I'm speaking of that are in the chromatin are called histones. They look like little beads. And these histones, they have a positive charge. So if these little circles are sort of like your histones, and you're going to see a picture of this on the next page anyway, and this is sort of your DNA, it sort of wraps around the histones, coils up around them, sort of like how you would put thread or yarn around a spool to organize it. And opposites attract, so the negative and the positive are attracted to each other. And that is what chromatin is. It's the DNA sort of loosely wrapped around these histones. There are some other proteins there that regulate gene expression. What does that mean? Well, remember, the job of DNA is to make proteins. When it's doing that, it's the genes or the codes for the proteins are being expressed. That's the terminology that they use. So certain proteins turn that DNA off and on. Because if you think about it, a blood cell and a bone cell and a skin cell all have the same exact 46 chromosomes. So then why don't they all look exactly the same? Because when the cells become specialized to do their special jobs, some DNA gets turned on and some DNA is turned off. So even though all 46 chromosomes are there, it doesn't mean all 46 chromosomes are being read. Only certain sections are being read. And so there are proteins that determine that basically turn the DNA on and off in different cells. Okay, here's a diagram to kind of show you. So if we look here at the bottom, this is your DNA, the double-stranded DNA here. And here they're showing the histones, how the DNA starts to coil, actually here, around these little bead-like things, these histones. And it wraps around them, sort of like that. And then these histones actually coil up too, and you end up with this is what your chromosome actually looks like. The DNA really tightly coiled. So a chromosome is not just this little short double helix, and because the chromosome looks like an X, it's two little short. It's not like this. That is not what's happening. What's actually happening when a chromosome forms is your DNA is basically coiling up to 50,000 times more compact than if you stretched it out. And that's why you can only see the chromosomes when the cell is dividing. Because if this was a set of instructions, you can't read the instructions when they're tightly coiled up like that. You could only read them when they're uncoiled. So the cell normally, you can't actually see the chromosomes. It coils up in chromosomes when the cell's dividing, and when the cell is not dividing, it sort of uncoils, and it's more loose, and it looks kind of stringy. Any parts that stay condensed means coiled up, are never expressed, meaning those parts of DNA that stay really tightly coiled do not actually code for anything. They're not actually read to code for something. And here's one more picture showing how the DNA is so tightly coiled up. You have your double helix at the top, coiling around the histones. Those coil up even tighter, and eventually down here at the bottom, here's your chromosome. And this little coiled section is just one little tiny bit of this chromosome. Okay, chromosomes are actually one strand of DNA, but when the cell goes through the cell cycle, which we're going to talk about tomorrow, the chromosome does make a copy of itself. And then it has two sides, and those two sides are called chromatids, or sometimes they're called sister chromatids. And those sister chromatids are connected to each other by this little section in the middle called the centromere. So this picture is showing this is one chromosome. So your cell, when your cell is first born, your chromosome would technically look like this. This would be one chromosome. But when the cell gets ready to divide, it needs to, how's it going to give a copy of its information to the new cell? It makes a copy of itself. Now, this is still one chromosome. But when the cell divides into two, this chromosome can split in half, and each new cell will now get a copy of the information. So that's why the chromosomes look like an X. Technically, you don't need the whole X. A cell that's never going to divide, its chromosomes wouldn't look like X's. Its chromosomes would actually look like sticks because it would have no need to make that extra copy of its information. So just to clarify. And here's another picture. This one actually has a spiky thing called the kinetochore on it, which you will not be tested on. But little fun fact, when the cell goes to divide and the chromosomes line up, you may remember learning about mitosis in freshman biology. What actually connects these chromosomes to the spindle so that they can get pulled apart is these spikes, the kinetochore. So located in this centromere, in this middle part, is this kinetochore, and it connects the chromosomes to the spindle fibers so they can get pulled apart during mitosis. But again, you won't be tested on it. Again, this is called a homologous pair of chromosomes, and we'll talk more about that later. This would be before replication on the left, and after replication, or when the chromosomes make a copy of their information on the right. This is still two chromosomes, and this is 2 chromosomes. These chromosomes have just made an extra copy of their information, but this is still called 1 chromosome, just to make sure that's clear. A lot of people get it confused. And this is showing it one more time, 1 chromosome. In step 2, the chromosome's made a copy of its information, and in step 3, the cell is now dividing, and each new cell is getting a copy of that information. So during cell division, the chromosomes become duplicated into chromatids, and then what will happen is during mitosis, as I said, those chromatids will separate. So if this cell had 4 chromosomes, here's what would happen during mitosis. They would line up, the cell would split in half, and each new cell would still have 4 chromosomes, but the chromosomes would be sticks instead of X's. Still 4 chromosomes. And these cells would be identical to the original cell and to each other, since these 2 sides of the chromosome are identical. They're sister chromatids. Okay, the last thing here for today is diploid versus haploid. Somatic cells, or body cells, and you really should know this word, somatic. Somatic. This is the word you're probably going to see. You're probably not going to see body cells. Somatic cells have 2 copies of every chromosome, and this is called the diploid number. It's represented as 2 with 2N is the symbol for the diploid number. Why do you have 2 copies of every chromosome? Because you got 1 from your mom and 1 from your dad. Remember, sperm has 23, the egg has 23. When they get together, the zygote that's formed has 46. And so they are in pairs, 46 total chromosomes in 23 pairs, because there was a chromosome number 1 and a chromosome number 2 and a chromosome number 3 in the sperm and in the egg. And we'll talk a lot more about that in the next chapter. Now, sperm and eggs have haploid number of chromosomes. Also, different species have different numbers of chromosomes, just so you know. And then, like I said, gametes, the sperm and egg, they have only half the number. They have the N number of chromosomes. So N is sort of like the base number, and then 2N is when there's 2 copies. Don't get mixed up. A lot of people get mixed up, and they think when I say 2 copies, I'm talking about this, that this is 2 copies. That's not what I mean. The pair of chromosomes that I'm talking about, these are called a homologous pair. They are not identical to each other. Chromatids are identical to each other because they're copied from each other. But if you have 2 copies of chromosome number 1, your 2 copies of chromosome number 1 are not identical. They have information about the same stuff, but they're not identical. And that's the whole idea of genetics. If you remember, somebody could be, for example, big B, little b. That's because one chromosome could be carrying big B they got from their mom, and chromosome they got from their dad has little b. So now these are genes for the same trait, but they're not, the 2 chromosomes are not identical. And hopefully this will clarify it. This is called a karyotype. This is a picture of the chromosomes. So this is a normal karyotype of a diploid cell. Notice how there's 2 copies of chromosome number 1, 2 copies of chromosome number 2. And in a karyotype, these are put together, they're matched up in pairs based on the location of the centromere, the bands, there's a pattern of bands on them, the size of the chromosomes, and the last pair is the sex chromosomes. This happens to be a male, so it's XY. The Y doesn't really look like a Y, but it's this little tiny chromosome. A female would be XX. This is on the right, a haploid cell. So for example, if the male on the left made a sperm, 1 copy of chromosome 1 would go to that sperm. It would be random which one it would be. 1 copy of chromosome 2, 1 copy of chromosome 3, and an egg would do the same thing. And then when the sperm and egg get together, you get a baby. Notice, by the way, since a female is XX, the female, the egg, can only have an X. And that means that the male is going to determine whether a baby is a boy or a girl. If he gives the X, and this sperm fertilizes an egg, you'll get a girl. If he gives the Y, then you would get a boy. The female will always give an X because she only has an X. Here's another karyotype stained with a fluorescent dye, and I just wanted to comment, 46XX or 46XY would be the way you'd write the notation for that. if this had an extra one, for example, let's say this had an extra 21, an extra chromosome 21, you'd have 47 XY plus 21, telling me it's number 21